in case, in case you care. And so we're going to turn it over to Shiv Pillai, who's going to tell you about B cells. It's
I have a mic.
Oh, you got a mic.
Yeah.
We'll turn it off to Shiv.
Okay. On a rainy April day in Boston in 1996, I was driving across the river and I had an epiphany. I was on my way to what I thought would be one of my more challenging teaching assignments. And I was going to be at an elite academic institution in Cambridge, where I was supposed to give a 45-minute lecture, sort of an overview of immunology. And I was sure the students would be very bright, but I was a little concerned that they might not have the required background to follow what I wanted to tell them. But when I reached the classroom, all my trepidations vanished. The students had this very sophisticated understanding of immunology. The average age of the class was 6. This was my daughter's kindergarten class. And all these kids had been brought up on a television show, which some of you might have watched, called The Magic School Bus. And by watching the show, they knew that white cells were the good guys and bugs were the bad guys, and that white cells could swallow up these bugs whole and chew them to bits, or they could shoot out these magic bullets, these cruise missile-like things called antibodies that would go and home in on their targets and blow them up into smithereens. Even more sophisticated understanding existed in this room. I realized that they understood that white cells attack bugs, but they don't attack our cells. This is non-self recognition. And they also were aware that if white cells saw a bug, and if the bug was terribly yucky, like a piece of brown bread, it could be coated with this antibody peanut butter and then become really delectable, and then it would be swallowed up. And this is opsonization. So immunology, whatever a bull might have told you yesterday, is actually very simple. It's just about white cells and antibodies. And we're going to talk about some of the white cells and antibodies in this lecture. So in the 19th century, just to go back to how this all started, so the second half of the 19th century is probably one of the most exciting periods in the history of biology and medicine. You have John Snow and the discovery of the Broad Street pump, then you have Darwin, you have Mendel, you have Metchnikoff. And then by the 1880s, most of the excitement centered around two powerhouse laboratories. You had the laboratory in Berlin of Robert Koch, and you had the laboratory in Paris of Louis Pasteur. And young men from all over the world, this was very much a male preserve still, would flock to these two labs, and the pace of discovery was truly remarkable. To cite just one example, in 1888, Loeffler described the bacillus that causes diphtheria. In 1889, Roux and Yersin showed that supernatants of this bacterial culture made a lethal toxin. In 1890, Brieger and Frankel showed that small amounts of this toxin could be injected into guinea pigs and create a form of immunity. And later that same year, Shibosaburo Kitasato and Emil Behring used iodoform to chemically modify this toxin and inject increasing amounts of it into larger animals. And they found a principle in the serum of these animals, which they called an antitoxin. And within 1 year of this discovery, kids who were dying, gasping for breath from diphtheria, were being miraculously saved by the injection of this antitoxin. So around this time in the laboratory, there was a young man who was really Koch's favorite, a brilliant guy called Paul Ehrlich. And Ehrlich is clearly the genius, along with Darwin, perhaps one of the greatest geniuses of biology. And it was clear that Ehrlich was different from everyone else. This is a guy who could barely get through medical school. He had to go to 4 different medical schools to get his degree, because he would never look at anatomy and things that other people were studying. He was busy doing experiments. Everybody knew that there was something special about him. So he was the kind of guy that would interpret your data if you had time to think about it. And he was somebody that Behring did not like very much. And so Behring comes out as this, slightly pompous, very correct, formal, very honorable, somewhat, Teutonic caricature of a professor. Whereas Ehrlich was this person who would be scribbling on your shirt, if it wasn't his own shirt, scribbling on the floor, coming up with ideas all the time. And he realized that these guys were onto something much bigger than just antitoxins. He realized that they discovered that if you injected something foreign, something he called antigens, you then produced a response and you made some kind of a molecule which he called an anticorpor or an antibody. And that these antibodies were then going to protect you from subsequent infection. So Ehrlich also came out with the idea of autoimmunity. We called it horror autotoxicus. He came up with the thought of tumor immunology. He described what tumor immunology would be all about. But his greatest contribution at that time, and it's worth thinking that this is the 1890s, it's 60 years before any polypeptide hormone receptor has been even thought about. It's about 70 years before we have a good model of the fluid mosaic theory of the membrane. And he came out with a model called the side chain hypothesis, where he said that immune cells would have antibodies on their surface, which would function as receptors. And when these receptors were triggered by antigen, the cells would then make more of the same antibodies. So this is a concept of a receptor-ligand interaction that was well before its time. In 1900, the first Nobel Prize for medicine went to Behring. In 1905, the Nobel Prize went to the Grandmaster Koch himself. And in 1908, Mechnikov and Ehrlich shared the Nobel Prize for their theories on immunology. And then for about 50 years, nothing much happened. So in the mid-1950s, we have McFarlane Burnett and Talmadge come up with this idea of the clonal selection hypothesis, where they argued that lymphocytes are basically individuals. I'll just figure out how this works. Yeah. And that if antigen comes by, antigen binds to one of these lymphocytes, the lymphocytes expand and proliferate, this clonal expansion, and then they go on to secrete antibodies. And it's clear from this view that if you thought about it, antibodies had to exist in two forms. A membrane-bound form that functions as a receptor, and a secreted form which provides protection. So in today's lecture, we'll talk a little bit about the function of antibodies. And I think you all know this from your high school days, but we'll just touch on it briefly. We'll talk about B-cell development. We'll talk about B-cell activation and a lot about the role of helper T-cells in antibody production. We'll talk a little bit about therapeutic targeting of B-cells, and then there'll be a summary. We know that the relevance of B-cell development and B-cell activation from a clinical standpoint, it's very important from the point of view of immunodeficiency syndromes, which you'll hear more about tomorrow. We all know that any vaccine that actually works today, anything that provides protection, does so by providing us with high-affinity antibodies. So without high-affinity antibodies going through the somatic mutation process, you're not going to get protection. We're going to recognize that B cells are extremely important in autoimmunity, not just in diseases that we think autoantibodies are important in, but in diseases that we think of T-cell origin, B cells play a very important role. We'll come back to that. And we won't talk much about this. This will be covered tomorrow by Steve. And allergies and atopic disorders, again, B cells are important. And then lymphomagenesis. 90% of all human lymphomas arise from B cells, and most of those arise from the germinal center. So looking at the germinal center reaction is quite instructive. And those of you who are following the field and looking at all the exome sequencing results coming out from lymphomas, there's a lot of connections that you can make with the biology that goes on there. So clearly some diseases, in some autoimmune diseases, autoantibodies are important. But I think what we'd like to stress, and I'll come to this at the end of the talk, that B cells are required to make CD4 memory. So all those T cell subsets you were hearing about yesterday, if they aren't really memory cells, they aren't really fixed. Some of them are pretty plastic. But memory cells are much more fixed. They're committed. And you need B cells to make CD4 T cells into memory cells. And this becomes really important in the context of autoimmunity. There's probably a role for B cells. Of course, there's a role in antigen presentation. We'll talk about that. There may be a role in cytokine secretion. There is a field where people study B-regs, and I'm happy to discuss these cells if you're interested in them. But I would tell you that I think right now B-regs are very much a phenomenon of 2 or 3 mouse models, and they are important, and their relevance in humans, there are cells which can make IL-10, which are B cells in humans. The actual relevance is not clear. So if you have questions, I'd be happy to discuss this. Okay, so again, this is just re-stressing. Antibodies are really important for defense. Antibodies mediate disease. Antibodies are used therapeutically. Okay? So, and for those of you who have memorized this kind of stuff in medical school, and if you divide diseases into type 1, type 2, and type 3, all 3 types of diseases depend on antibodies. Whether you're dealing with allergy-like symptoms or dealing with antibody-mediated diseases where antibodies are against tissue antigens or cells and so on, or if you're dealing with immune complex disease. So all of these are antibody-mediated. So a lot of pathology is mediated by antibodies. Okay, so I know this is a repetition, but I'll just go over it very briefly. The structure of an antibody is this Y-shaped protein, and you have variable domains for the heavy chain and the light chain, shown here as V's. And it is in these variable domains that there are 3 regions, which we'll talk about, which are very different from one antibody to the next. And we call them hypervariable regions or complementarity determining regions. And it is the association of these hypervariable regions of the heavy chain and the light chain that creates the combining site where the antibody, so yellow is the light chain, red is the heavy chain, the heavy and the light chain, using those hypervariable domains, recognize antigen. Okay? It's worth keeping in mind, just from a brief structural standpoint, that you should think of each of these domains, each of these antibody domains, as being a sort of a ribbon-like structure, beta strands, forming a bunch of beta strands forming a spread-out ribbon. And then if you took this spread-out ribbon and folded it together, and you clipped it in the middle with a disulfide bridge, you would create what's known as a beta barrel or an IG fold. And this is the basis of an antibody structure, but it's something that exists in evolution from archaebacteria onwards as a domain in proteins. But when you make the structure, what I would like to emphasize is that between these beta strands, which you'll see in the next figure, there are some loops. Okay? So these are these strands, the beta strands. If they were folded out and then brought together, you would have this beta barrel structure. But these things that stick out like fingers on the top, these are those loops which bind the antigen. These are complementarity determining regions or the hypervariable regions, and they're not part of the real basic structure. They're part of these loops that stick out and are going to be different from one antibody to the next and will create the site for recognition of antigen. Okay? You should think of the T cell receptor in the same context. I'll come to that in a minute. So if you looked at the sequence of a bunch of light chains, for instance, you would notice that there was a most variability in 3 regions, which are CDR1, CDR2, and CDR3, shown over here. And those are these 3 loops that stick out at the top of this barrel. Okay? This is the most variable part of the antibody. And if you were thinking of the entire antibody, you would think of 3 fingers coming from the heavy chain, 3 fingers from the light chain. These are the hypervariable loops, and they would come together to create a site. If the antigen is a small molecule, like some sort of a hapten over here, then these loops are going to come close together and form a cleft. If the antigen is a protein or something larger, the loops are going to spread out and form a surface. So you see that over here. They form some sort of a surface, and the antigen binds to the surface. If you think about the T cell receptor is something in which the loops play out and form a surface, and that can accommodate MHC and peptide. So it's very similar to an FAB of an antibody. So again, this is just a picture of a model of the FAB part of an antibody that recognizes a protein. The protein is N-glycosyme. All the residues here in red are from those hypervariable loops, and they're involved in recognizing the red residues in the antigen. This is a glutamine shown over here in purple, which fits into this little cleft that you see in the middle over here. So basically, there's a complementarity, and the complementarity is created by the hypervariable regions, which spread out, are different from one antibody to the next, and provide the recognition basis for antibodies. So as you know, antibodies come in different flavors. You have IgG, IgD, IgE, IgM, which is pentameric, and IgA, which is a dimer, at least in secretions. So these antibodies all have different functions. We'll touch upon them briefly. I just want to emphasize that the multimeric antibodies have a J chain. So you have this covalent association of these tetramer units into this pentameric structure, and then you have association with the J chain, and the J chain is what is going to be recognized, and you'll hear more about this tomorrow or whenever Rick talks, and it's involved in recognition by the poly-Ig receptor for transport across an epithelial surface. So whether you think of the lung or the reproductive tract or the GI tract, antibodies containing J chains can be transported across surfaces. IgA, of course, has a J chain as well. Okay, so the effector functions of antibodies, which are summarized in a textbook which is not being given to you if you win the raffle today, is over here. So one function, of course, is neutralization. Antibodies coat the bug. The bug, maybe the receptor for the virus to enter the cell is now occluded, and therefore the virus can't enter the cell, and that's called neutralization. Same thing could happen with toxins. If you coat a bug to make it interesting enough for a phagocyte to take up through an Fc receptor, that process is called opsonization. So the antibody coats the bug, the Fc portion of the antibody sticks out, is recognized by an Fc receptor on the phagocyte, and that's opsonization. Sometimes an NK cell can recognize an antibody that coats a target cell. The antibody that coats the target cell activates the NK cell, and I'll describe this in a later slide, and this process is called antibody-dependent cellular cytotoxicity. And then you have complement, which can lyse microbes, it can provide opsonization, and it can also drive inflammation, and I'll describe this again in a later slide. Okay, so complement is summarized in one slide over here. So basically, the antibody binds the tissue target. When it binds the tissue target, it can fix the first component of complement. If complement is activated, there is a C3 convertase that is generated, which cleaves C3 to C3b and C3a. C3b can be covalently attached to a microbe and serve as an opsonin, because we have receptors for C3b on many of our cells, on phagocytes. So basically, one function of the antibody here is opsonization. Once you make C3, that helps make a C5 convertase. The C5 convertase cleaves C5 into C5b and C5a. C5a is an anaphylatoxin and a chemokine. It can drive inflammation by bringing in white cells to the site of infection. And so this is a driver of inflammation as a chemokine, whereas C5b nucleates the formation of the C5 to C9 complex. C9 forms the pore. The pore can make holes. Bacteria then have these holes, and their stuff can extrude out of the bacteria, and they can be blown up in this process. Okay, so that's complement. Stop me at any time if you find you're very interested in something, you want to talk more about it. I assume it won't be complement. I'll talk about this very briefly. The poly-Ig receptor it's an endocytic receptor which mediates transcytosis, not endocytosis, but transcytosis. So it can bind a J chain containing immunoglobulins. In the lamina propria, you might have an antibody secreted with a J chain on it. The poly-Ig receptor binds to it, internalizes it, doesn't take it to a lysosome, takes it from the basolateral surface to the apical surface. There, the poly-Ig receptor is cleaved. The cleaved extracellular domains form what is called the secretory component, and the IgA is secreted and better protected from proteases by the fact that it's associated with the secretory component. Okay? I won't belabor this. Ig is bound to its receptors on mast cells and so on. So it's a high affinity receptor. And if an allergen comes by and is multivalent, it triggers the Ig, the mast cell gets triggered, it releases all sorts of stuff, and then you sneeze and you get an allergy or you get anaphylaxis, and that's basically the whole component. I've summarized Steve's lecture for you in one slide. Okay? Now, Abul alluded to this. He said that it's often useful to make an Fc fusion of some receptor or extracellular domain of a receptor to prolong its half-life. So do you know where most proteins in the blood go? Most of your serum proteins. They actually don't have very long half-life. Where do they die? Does anybody know? Yes, somebody said it. Liver. And why did they go to the liver? Say that again? Not exactly. No, they get broken down, all that happens, that's true. But why do they get into liver cells? Because of glycosylation, that's true. So there's a receptor on liver cells, which is the asialoglycoprotein receptor, which swallows up all our plasma proteins, and they get chewed up and they're used for other things. But basically, all serum proteins are rapidly turned over because they're taken out by a receptor which binds the galactose residue stuck on the sugar side chains. But IgG has a special receptor called FcRn. So IgG gets into endothelial cells, is bound to the FcRn. So basically, it goes through a sort of a rest and recreation period inside endothelial cells. So instead of going through the blood and getting swallowed up by liver cells, it hides out. IgG antibodies they hide out in these endothelial cells because they're internalized, and then they're slowly released back into the serum. So in this way, their half-life is prolonged just by keeping them subendothelially for a while in endothelial cells and releasing them and taking them up and releasing them again. So when they're needed, they're still around, and they're not being rapidly cleared by the asialoglycoprotein system. So this mechanism makes IgG a particularly long-lived protein. Its half-life is about 3 weeks. So if you take anything else and attach it to the Fc portion of IgG, it becomes a long-lived protein. And that's one thing that Big Pharma uses to connect proteins to IgG in order to make them live longer. Okay, so IgG, so FcRn is a receptor for transcytosis, for endothelial internalization, and so on. But the receptors that trigger inflammation typically are Fc receptors, like Fc gamma R1. And on our macrophages and neutrophils and so on, we have activating Fc receptors, which have ITAM motifs. These are tyrosine-based activating motifs. And to oppose these receptors, which are going to drive inflammation, they're going to drive opsonization and inflammation, clearance of microbes, and so on, they're opposed by an inhibitory receptor, also found on most innate immune cells, and also found on B cells, called Fc gamma R2b1. And Fc gamma R2b has an ITIM motif. It's an inhibitory motif. It sends inhibitory signals, and it can dampen the inflammation. So it's sort of a yin-yang process that goes on, where IgG molecules, they're going to drive inflammation. It's good for you to some extent, but too much inflammation is not good, so you have to dampen it, and you have inhibitory receptors that drive the reduction of inflammation. And these receptors on innate immune cells become quite important when you try to treat diseases with IVIG, which we'll come back to. Okay, this is the last slide, I think, on antibodies. So just to remind you that NK cells have a particular type of Fc gamma receptor, so CD16, which can recognize IgG molecules which coat an infected cell. So this is a virally infected cell. This is your viral antigen, and you have antibodies coating them. The Fc receptor on NK cells can be triggered. The NK cell is then activated. It releases perforin and granzymes that you heard about yesterday, and these can then cause the apoptotic death of the infected cell, and this process is called antibody-dependent cellular cytotoxicity, or ADCC. And it may be even more important than we think. It may be relevant beyond just the clearance of virally infected cells. Okay, and just to close this section, we have Fc gamma R1. This is a high affinity receptor for IgGs. Fc gamma R2b, I showed you this one here, is the inhibitory receptor with the ITIM motifs. And this guy over here, Fc gamma R3a in humans, is the activating receptor. And this is the one that's present on NK cells and drives NK cell activation. Okay, I'm going to summarize this antibody portion of the talk by saying antibodies protect you, but antibodies kill. Autoantibodies can make you very ill. Antibodies coat pathogens. They coat the infected cell. With NK cells and phagocytes, they give those microbes hell. When antibodies fix complement, the shit really hits the fan. You'll be blown to bits, little microbe. run away if you can. You take a set of beta strands, you get a beta pleated sheet. Fold the pleated sheet in two, and the barrel is complete. Clip it with a disulfide bond, and you have an IG fold, found even in archaebacteria. This domain is old. Loops make connections between the beta strands. If you want to make an FAB, please use both your hands. The loops are like fingers. They stick out at the top. Complementarity determining regions, the cream of the crop. 3 fingers from the heavy, 3 fingers from the light, come together to create an antigen binding site. Bring the fingers close together and you have a cleft with a purpose. Splay the fingers widely and you have a protein binding surface. Antibodies protect you, but baby, antibodies kill. Autoantibodies can make you very ill. Or you Y-shaped globular proteins with the name that Ehrlich gave. If the good doctor could hear this doggerel, he'd be turning in his grave. Okay, thank you. I'll take a pause. Thanks. So I'm going to talk now about B-cell development. And I want to remind you, it's actually useful to be somewhat polytheistic when you think about immunology. There's actually no real difference between B cells and T cells. And I like T cells as much as I like B cells. There's a close symmetry between everything that happens in B cells and T cells during their development. And even we'll come to it functionally as we go on in this talk. So first, I just want to point out there's a symmetry between B and T cell development. You have a hematopoietic stem cell, and the stem cell gives rise to some common lymphoid progenitor. The progenitor then gives rise to a committed B or T lineage cell, which are going to do very similar things molecularly with the antigen receptors. They're going to first commit and start rearranging the antigen receptors. So you get a B cell or a T cell in that process. Then they're going to go through a checkpoint, which I'll describe a little bit, which is based on reading frame. So when you join 2 pieces of DNA together, and that's how we join our antibody genes and our T cell receptor genes, you could decide to do this perfectly. And we are capable, fully capable of joining 2 pieces of DNA, which we do all the time, perfectly, with a perfect junction. But when we make our antibodies and our T cell receptors, we decide to do this with the addition and removal of bases at our junctions to create more diversity. And if you add bases between 2 bases of DNA, and you don't add them in a multiple of 3, if you add 2 bases or 5 bases or 8 bases, you're not going to get the right number of amino acids that will keep the whole thing in frame. So we need to have a selection event while we create diversity, using this tool of just making rearrangements with great junctional diversity. We need a selection event where you select for reading frame. And I'll talk a little bit about that. It's linked to disease in humans. It's linked to disease for T cells as well when you think about it. But it's something to keep in mind. Once that is done, that selection event, which I've called here as positive selection 1, is connected to the process of allelic exclusion. Allelic exclusion means that you want to make sure that your B cell has only 1 antibody receptor, or your T cell has only 1 receptor for antigen. Okay? And that's part of this process which we'll talk about. Subsequently, you go through what you would conventionally call positive selection in T cells, where you make lineage commitment events, and you select cells so that they match the right co-receptor in the case of T cells, and you actually create different lineages in B cells. And you also have negative selection events, which get rid of self-reactive cells. So all these things happen both in B cells and T cells, and I'll describe this from the B cell context, but if you have questions about T cells, we can address them at the same time. Okay? So in the case of commitment to the T lineage, notch 1 is important. In the case of the B lineage, we actually understand quite a bit about the transcription factors that drive the process. Again, if you have questions, I'd be happy to deal with them. Okay? So there are 2 lineages. Like we have alpha, beta, and gamma, delta T cells in the T lineage, there are 2 B lineages. There is a B1 lineage that arises from fetal liver cells. And like gamma delta T cells, B1 cells have a restricted repertoire. They can see only a certain number of things. There was a lot of doubt whether B1 cells existed in humans, but over the last 2-3 years, there's enough data to address this issue, and we now accept that humans also have B1 cells, okay? And that these B1 cells are these fetal liver-derived cells, at least in mice. We don't know that in humans. And these cells have this restricted repertoire. In mice, they have CD5. In humans, they don't. The other major lineage is, of course, the B2 lineage. And these are real lineages, okay? These are committed cells. Once they're committed, there's no plasticity. Once they go, they're completely done. So the B2 cells can give rise, just like you get CD4s and CD8s from alpha beta T cells, at the end of the day, B2 cells can give you marginal zone B cells and follicular B cells. The follicular B cells sit in follicles, talk to T cells. The marginal zone B cells are very long-lived cells, deal with lipid antigens. They have slightly different functions, okay? So B1 cells, they have fetal liver progenitor. They are a major source of mucosal IgA, okay? So these are mainly at mucosal sites, the peritoneum, pleural cavity, other mucosal sites. These cells are a very major source of mucosal IgA. They also, some of you may have seen this paper in the last few months, they're also the main cell in the body that makes GM-CSF in response to LPS. Okay? So there's a relevance to septic shock. So just keep in mind, some B cells, so normally GM-CSF is made in the bone marrow by stromal cells. But if you trigger toll-like receptors, the source of GM-CSF is B1 cells. So there's an interesting science paper addressing this recently. So just keep in mind that some B cells make cytokines. And GM-CSF, if you trigger cells, is going to be made by your B cells, by B1 cells. Marginal zone B cells, like B1 cells, are self-renewing. That means they have stem-like properties. They live forever. They deal with blood-borne pathogens. A lot of T-independent responses stem from them. We'll talk about that. They might be capable of doing instant T-dependent-like responses with NK T cells. So NK T cells are these innate-like cells of the adaptive immune system. And marginal zone B cells can be thought of innate-like cells of the humoral immune system, also adaptive, and these can interact with each other and you can have activation. They're very important for lipid antigens. Jason's sister has shown that they can be relevant to shuttling immune complexes to follicles. Okay. Now, I'm going to tell you this as though this is a textbook view. So most textbook views are wrong. So just assume that this is broadly correct, but there's no absolute here. So we say most follicular B cells are the ones that give rise to high affinity antibodies, isotype switch cells, and long-lived plasma cells. These are the ones that mainly give you protein-dependent, T-dependent responses. And we'll talk a lot about this. But that doesn't mean that marginal zone B cells can't do that. Okay? So marginal zone B cells mainly respond to polysaccharides and lipids. B1 cells mainly respond to polysaccharide and lipids. So we generally think of these cells mainly being T-independent and these cells mainly being T-dependent in their responses. But this is not absolute. Okay? And there are enough exceptions for you to think about. But generally speaking, this is true. Okay, so there are a number of checkpoints during B cell development. I will spend a little time on this checkpoint where the pre-BCR plays a role in selection. So just to recap for you, during commitment, you start rearranging your immunoglobulin heavy chain gene. You rearrange from a D to a J, and then you go through the next step, which is a V to a DJ rearrangement. When you make this rearrangement, 1 in 3 cells roughly will do this right. 2 out of 3 cells will screw up because they added the wrong number of bases. Those cells deserve to die, and they will be allowed to die. The guys who do the right thing will be selected and told that they've done the job properly, and they will be allowed to expand tremendously. You'll have the biggest expansion of the B lineage at this point. And we'll talk about a human disease that is linked to this in a bit. Okay? Subsequently, at the immature B cell stage in the bone marrow, you do have selection based on self-reactivity, and we'll talk about that. And then once you go out into the periphery, actually we continue to have, in B cells, you don't finish everything in the central organ. B cells go out to the periphery and they are still immature at one point. They're called transitional cells, and they can still be eliminated or corrected if they're self-reactive. Okay, so self-reactivity, we can correct it in immature stages. Then they mature into, say, follicular B cells or marginal zone B cells. Yes? So this is the rule. When you make a B cell, It goes from the bone marrow as an immature cell only to the spleen, unless you remove the spleen. If you remove the spleen, it'll just find its way somehow into a lymph node, because maturation can occur in the bone marrow. Okay, but normally, at this point, the cell is going to leave and home to the spleen, enter the follicle in the spleen, change its phenotype, get IgD on its surface, and now acquire the ability to become a recirculating cell. And a recirculating cell is one that goes from lymph node to lymph node to lymph node, searching for antigen unless it dies of unrequited love. Okay, so that's the property that is acquired in the spleen and the follicle. So once the cell gets IgD on its surface and CD23, it now has the ability to recirculate, which means it's going to just move on from one node to the other. It can go back to the bone marrow as well, but keep recirculating. Okay? Whereas these guys can only go to the spleen. Okay? All right, so I told you that there was this checkpoint over here, where if the cell did the right thing and it made a heavy chain protein, it was in frame, it could make a heavy chain protein, it is going to assemble a structure called the pre-BCR, and this is a positive selection checkpoint. So the pre-BCR looks like this. It has a heavy chain. So this is if the cell made the heavy chain protein because it went through a good rearrangement. And the heavy chain will assemble with these pre-existing chains, which I described a long time ago, called surrogate light chains. So lambda 5 and VPB are surrogate light chains. They're sitting around in the cell. The cell that can rearrange the heavy chain properly will assemble this receptor. When it assembles, and it assembles with some signaling proteins called Ig alpha and Ig beta, which we'll come back to. And this assembled receptor, this is not a receptor that is designed to monitor the environment. It is a receptor that is monitoring the cell to see if it made the right reading frame and the right rearrangement. So the moment it assembles, it goes into the on mode. And this is understood a little better today. And it doesn't need a ligand. It's on when you assemble it, and it signals to the cell and says, you did the right thing, you can expand, you survive, you can expand, you can shut off rearrangement at the other allele, and you can then proceed to light chain rearrangement. Okay, so this is a ligand-independent signaling event, which occurs when you assemble the receptor. Same thing happens in T cells with the pre-TCR, and it basically identifies cells that made good rearrangements at the heavy chain. Okay, question? Yeah, so I don't know if I was slide. Oh, I have it all here, sorry. This, you can read this in your slide, but I'll explain it to you. So heavy chain rearrangement is a relatively slow process. Okay, so you do D to J, then you do V to DJ. When you make the first V to DJ, if it is in frame, if by some chance it's in frame, and it makes the heavy chain protein, soon after that heavy chain protein is made, a signal is delivered, and the other heavy chain locus, which might have been getting ready to do the V to DJ rearrangement, is blocked. So at the chromatin level, it's just wrapped up. So you cannot make the rearrangement of the other allele. If the first rearrangement fails and goes out of frame, doesn't make a protein, then the second allele goes ahead and makes a rearrangement. So the signaling shuts off rearrangement of the other allele, and it also shuts off expression of surrogate light chains, because now the cell has done the right thing, it doesn't need surrogate light chains anymore, and it induces rearrangement of the kappa light chain, the conventional kappa light chain. And it's always kappa first. Okay? So signaling through the pre-BCR, you have inhibition of heavy chain recombination, it's called allelic exclusion, you have proliferation of these B cells, you have stimulation of kappa light chain rearrangement, and you shut off the expression of surrogate light chains, again at the chromatin level, all through this signaling. Okay? So this is just to tell you that this checkpoint, this pre-BCR checkpoint, is defective in X-linked agammaglobulinemia. So in Bruton's disease, boys are born with no B cells, and they're born with no B cells because a signaling protein downstream of the pre-BCR, that's BTK, can't signal. And this signal normally gives you the biggest expansion of the B lineage. So any cell that's done the right thing, has made a good heavy chain protein, it expands like anything, and each progeny cell will then go and rearrange a different kappa light chain. And that's how you create your repertoire. You go through this process, you select the good guys, and then you let them expand, and each progeny cell rearranges a different light chain. Okay? Yeah, sure. Which leukemia? So, AML is a stem cell leukemia for myeloid cells, but if you were thinking of, there are pre-B ALLs where the transformed cell is actually a pre-B cell. And the signaling through the pre-B receptor, through the signaling machinery of the pre-B receptor, is actually very important in some of the pre-B ALL patients. And mutations have been found in SLP76, things that are really relevant to pre-BCR signaling. So almost all our leukemias and lymphomas, we use signaling processes that are normally used for proliferation, suborn them, and cause a cell to proliferate uncontrollably. Yeah? So from that one clone, I'm describing one clone, right? That one clone, let's say it multiplied and give you 1,064 cells, right? Each one of those cells will have the same heavy chain, but different light chains. So you'll have a lot of cells which have the same heavy chain, but different light chains. And you do this again and again, clone after clone, and create your repertoire. Yes? Okay, so the view, and this is a view actually we propounded about 20 years ago, and there's a lot of data supporting it, is that the pre-BCR will spontaneously signal. And recently, Hassan Juma, last year from Freiburg, showed that the pre-BCR assembles as a multimer because of an interaction of the surrogate light chain, of part of the surrogate light chain, with the sugar on the next heavy chain. And it assembles into an oligomer. And this oligomer, and there's a crystal structure from Chris Garcia that shows you that the pre-BCR is an oligomer. So I think the moment you make the pre-BCR, I mean, the term ligand is kind of probably, hard to define here, because it's the moment you make it, it doesn't need anything else. It assembles into an activated state. Okay, and that's something to keep in mind. It's not trying to look at antigen. It's just trying to see whether it was made. If it was made, it assembles. Yes? We're coming to lambda. Lambda is after a few slides. So you always first go to kappa. Okay? All right, so now we're talking, yes?
Is it a totally random mutation, or is there some pattern of mutation?
What mutation are you talking about?
Heavy chain.
So we are not talking about mutation at all so far, okay? We're talking of rearrangement. I take one piece of DNA and join it to another piece of DNA. We'll talk about mutation later. Okay, so this is a joining process where you join two pieces of coding DNA together, and it's a cassette model, where if each of us is a different B cell, we use different cassettes. We have the same cassettes in our genome, but we each have used different cassettes of V's and J's and join them together to get different antibodies. Okay? So what makes cells different is only what rearrangement event occurred, which is generally pretty random. It's not totally random, but it's pretty random. Okay? We can talk about that. It's not totally random, but it is pretty random. Okay, so now I'm coming to the question of kappa, lambda, and tolerance. Okay, so I'm going to tell you that the main mechanism of tolerance in B cells, and that B cells are actually much more politically correct than T cells. Okay, so in T cells, if you have a self-reactive cell, you kill it. In the case of B cells, if you have a self-reactive B cell in the bone marrow, and it sees your blood group A, it sees your blood group A antigen, it gets strongly triggered. Instead of killing it, you give it another chance, you let it make another rearrangement, and maybe get rid of the bad light chain, and make a new light chain, and this process by which you might change the receptor, if it's self-reactive, is called receptor editing. So about 75% of your B cells in the bone marrow start out self-reactive. If they're strongly self-reactive, activation of these B cells can induce this process called receptor editing, and you can also have as a backup process deletion. Okay, so you edit cells, but if you can't edit them, you can delete them. There is another process called anergy, which occurs even in the periphery. In the case of B cells, anergy really means that if you have a soluble self-antigen, which is not multivalent, doesn't trigger the receptor like crazy, but triggers it with high affinity, repeated stimulation makes that B cell very unhappy. It requires much more BAF to survive, and it can't compete with its neighbors, and it dies a slow death. and we call that anergy. Okay, so central tolerance in B cells, and I'll go over this a little more, receptor editing, deletion, and anergy. Okay? Now, receptor editing, to think about it, here you have a self-reactive B cell, which happened to have an orange kappa light chain. Okay? If it had an orange kappa light chain, and it could see self-antigen, let's say it could see blood group antigen really well, this B cell would be triggered. Triggering it would cause rearrangement of a yellow light chain, and the yellow light chain might be another kappa, or if all kappas failed, it could be a lambda. So you go to lambda only when you have exhausted your kappas. And all lambda light chain containing B cells are hallmarks of receptor editing having occurred in that cell. Okay, so the cell switches its light chain, hopes that the new light chain is not self-reactive. The edited B cell is no longer self-reactive, goes out of the bone marrow, goes to the spleen, and matures. Okay, so this is, I would say, the most important mechanism of central tolerance in the B lineage. Okay? And if you go through all the experiments that have been done over the last 2 decades, actually all add up to supporting this model, even though we've thought otherwise. Yes? No. Everything you do in terms of rearranging your heavy and light chain is to create diversity in your repertoire, right? Anything, but this is to get rid of self-reactive guys. So what happens is if the B cell here saw self, then it's not compatible with further life. So you have to either kill this B cell or change it. And what you typically do is to change it. You induce the RAG genes to which cause rearrangement. They rearrange the light chain gene again. And if they make a new light chain, the cell has got rid of the orange light chain, it now has the yellow light chain, now it goes out to the periphery. So it's really, the generation of the repertoire is huge, and it can be self-reactive. Subsequently, you cull it. B cells edit and they also die, and the T cells, though there is no evidence for editing, it is probably because nobody in the T cell field has done the right experiment yet. And there may not be. You have to make knock-in T cell receptors, which just has not been done. I can't understand why, but it's not been done. It should be done. It is likely that there might be receptor editing, but so far there is no strong evidence. There's one group, one person who's suggested that receptor editing exists in T cells, but the evidence is very weak and doesn't support this kind of a model. But you have to do the right experiment. I could explain this to you later. If you do a transgenic T cell receptor, it is useless. You need to make a knock-in because you have to get rid of the old T cell receptor every time if you want to test for editing. Okay, but not been done. Yeah.
If you look at, forget the knock-in and the transgenic,
Yeah.
just look at normal lymphocytes in a normal healthy individual. A lot of the B cells contain previously rearranged light chain genes.
If you search for them.
If you look for them,
Yes.
that's actually never been found.
But also not been looked for. People have found circles in T cells and thought these are early cells. And you don't retain them. Once a cell matures, even a B cell, once it matures, it loses the circles. So you can't look for it once they've matured. You have to look early. And you find them in T cells. You know that the alpha chain rearranges frequently. Now, is that part of editing, or is it just some junk rearrangement event? That's not clear.
I mean, I don't want to get for you guys don't into the subtleties, but just the important message that Shiv is giving is that if you look at your B cells, almost half of them have previously rearranged light chain genes.
Actually, the data from Michelle is about 75% is in humans. So there's a lot of rearrangement that occurs in humans. And in humans, there are a lot of lambdas, by the way. Mice have very few lambdas. Humans have a lot of lambdas, and all of them you can assume have gone through receptor editing. Okay? So I'm not sure about T cells, but could be. Now, evolution also, you have to, when you think about evolution of T cells and B cells, there's no reason, we can't understand why they evolved so similarly. I mean, what were the pressures that made them do things so similar? Sometimes they're obviously going to be different. Not everything's going to be the same. Now, what happens in receptor editing? Yes. Sure. Actually, heavy chains only in experiments gets edited. In real life, probably not. Okay? It's very hard to edit a heavy chain because you have 3 segments, V, D, and J. And it's just not an efficient mechanism. Same thing in the T cell receptor. The beta chain has 3 segments. So if you're going to edit, it's easier to do the light chain or the alpha chain. Okay, so the process of editing looks like this. The kappa light chain locus, you have a bunch of V kappa segments, and you have a bunch of J kappa segments, big distance in between. This is the constant region of kappa. You want to create a VJ exon, right, which is going to be heavy, the V kappa 29 here joined to one of these J kappas. Once you've created this in a B cell, let's say this particular VJ exon, which is going to splice to the constant region of kappa to give a kappa light chain, let's assume that this is self-reactive. So receptor editing means you take something upstream of V kappa 29, and you then go to a J kappa downstream of this particular J kappa, maybe to J kappa 5, and then you delete out the old VJ exon, you create a new VJ exon, you have a new light chain. And you went from orange to yellow. Okay? I mean, that's the basic process. And if this fails, if you do it on both chromosomes and you screwed up, you then go to the lambda locus and you try. Yes? Say that again? That's an exon. So when we join 2 pieces of DNA together by recombination, and you still have a spliced donor site at the end of the J, each J. Okay, so that VJ functions as an exon. It has a spliced donor site, there's a spliced acceptor site here, and you can splice at the RNA level. But at the DNA level, this is an exon. Okay?
Is it happening in the liver and the B cells at different locations?
No, in the liver, nothing happens. In the liver, V's and J's are far apart. Only in B cells, you rearrange them, right? I mean, this is just to tell you that in the germline, it's like this. In the rearranged B cell, you've put them together. Okay? Okay, so you probably know that we have primary responses and secondary responses. The primary response, you make some antibody, largely IgMs. The secondary response, if it's a protein antigen, you make more antibodies, higher levels of antibodies, and more IgGs. Okay, so there's a primary response, secondary response. IgMs usually mean recent infection. IgGs usually mean an older infection. That's just one way to think about it. Okay? So antigen can come to a B cell in many ways. When you want to activate a B cell, antigen can be delivered through an afferent lymphatic. It can be captured by subcapsular sinus macrophages and taken to follicles. Antigen can come through conduits. If it's less than 70 kilodaltons, for instance, there are conduits that come from the subcapsular sinus to the follicle, so the antigen can come directly. Antigens can come through the subcapsular sinus, go to the medulla of the lymph node, be taken up by real DCs, and DCs might then carry the antigen to the follicle as well. So there's many different ways in which an antigen may reach its B cell and activate it. Okay? And so in T-independent B cell activation, we think of signal 1 being cross-linking of the B cell receptor. I'll talk about that in a minute. And signal 2 coming either from toll-like receptors, because you have a bunch of toll-like receptors on B cells, so the pathogen has a toll-like receptor, so it's going to activate them, or using CD21, which can function both as for giving you a second signal and also as a co-receptor, and I'll describe that in a minute. Okay? So when you have T-independent B cell activation, typically you have limited isotype switching, typically no affinity maturation, and you have short-lived plasma cells. Okay, so T-independent antigens, no T cell involved, it's not a protein antigen, this is what you're going to get. So the way to think about it is that if you think of an antigen that is monovalent, like a monosaccharide, it can bind to the B cell receptor, but it can't create a signal, because it can just bind, but it can't do anything to the receptor. Okay? If I took this monosaccharide and I made it into a polysaccharide, so I have repeated units of this shape. Okay? This polysaccharide can bind this different B cell receptor molecules on the same cell, cross-link them, bring them together, and induce signaling. So a simple analogy would be that if I was a one-armed bandit, I can catch Steve, but then if I actually want to catch Mark, I have to drop Steve and then go and catch Mark. Okay? Or if I want Lou, I have to drop Mark and then catch Lou. But if I was a deity and I had 10 hands, then I could grab 10 of you together, bring you all close together, and maybe you'd make music. Okay, so a multivalent antigen can cluster receptors and provide a signal. A monovalent antigen does nothing. Okay? So this leads us to some definitions. Haptens are small molecules that can't activate the immune system. They are antigens, but they are not immunogens. T-independent antigens are usually multivalent. They are polysaccharides or lipids or nucleic acids. And T-dependent antigens are always proteins. Okay? So haptens, as by definition, small molecules. They are antigens, but they are not immunogens. All immunogens are antigens, but all antigens are not immunogens. Okay? So that small molecule, that sugar, is not an immunogen. So just to summarize B-cell receptor signaling in a quick way, imagine you have the B-cell receptor, heavy chain and light chain, associated with Ig alpha and Ig beta. Ig alpha and Ig beta have these tyrosine-based ITAM motifs. There's an associated signaling molecule, a Sark family kinase. If an antigen comes by and clusters the receptor, the kinase gets activated. Those ITAM tyrosines get phosphorylated. You have these bulges appear on the inside of the membrane. This can recruit a downstream molecule called Syk. In T cells, this would be ZAP70. So the Syk is now recruited to the receptor, it changes conformation, gets more phosphorylated, gets activated, and all of B-cell signaling flows from this. So basically, clustering the receptor, phosphorylating the ITAMs, allows you to recruit the downstream kinase, which is Syk, and the activated Syk gives you all the pathways downstream. Okay, this is explaining what the co-receptor in B-cell does. So imagine that you have an antigen, and the antigen is bound to a complement fragment. It's been opsonized by C3b, which is then converted to C3d. Okay? The antigen can be recognized by the B-cell receptor. C3d can be recognized by the complement receptor type 2, CD21, also the receptor for the Epstein-Barr virus. The CD21 molecule brings with it signaling molecules. The receptor is in a complex and brings signaling molecules with it. So once you cross-link both these guys together, CD21 allows the activation of the B-cell receptor because it brings kinases which phosphorylate the ITAMs. So signaling through the B-cell receptor is about 1,000 times more effective. So you get a much better antibody response if your antigen is coated with complement. So a complement fragment here represents innate immunity being activated, binding to the antigen, and this combination, it could be an antigen-antibody complement complex or an antigen-complement complex, allows better activation of the BCR. Okay, so you get stronger B-cell receptor activation because of complement fragments. Yeah? Yes? Yeah, it's just because it's artistically easier to show this. No, hold that thought. Okay, so let's assume you're right in thinking that for a multivalent antigen, correct? But let's assume this is a protein antigen, and it's not multivalent. This is a microbe drawn here, but just assume it's a protein, and it's not multivalent. Then it would be T-dependent. Then it would become T-dependent. I agree. So, but you could have a B cell that uses a protein antigen and complement to get activated without being clustered. We'll come to that. What I've drawn here shows it almost like it's a single receptor, but yes, assume multiple receptors are being clustered and multiple complement receptors are being brought in and it's being activated. But hold that thought and keep in mind that with the protein antigen, maybe with complement some signaling can happen. Okay? Because you could have this in one molecule. You could bring one CD21 complex and activate one B-cell receptor with one protein antigen. Okay? I hope I haven't confused you with this. We'll come back to that. All right, so this is a very good segue to T-dependent activation. I said T-independent activation, but now we're going to talk about T-dependent activation. And when we say T-dependent activation, I want you to remember two things. One is there has to be some activation of the B cell by the antigen to begin with, which doesn't involve the T cell yet. Okay? T cell is really important here, but to start the process, you have to identify the B cell, which is going to get T cell help. Okay? So I want you to just think about that. So I'll come to that here. Okay, so the consequences of T-dependent activation are, one is that you're going to get a low affinity receptor become a high affinity receptor. This process, which involves somatic mutation, is overall called affinity maturation. You're going to change the receptor from the membrane-bound form mainly on the B cell, mainly to the secreted form in a plasma cell. And finally, you're going to have to change maybe the isotype. This might have been an IgM or an IgD, and you might want to go to IgE or to IgA. And that process, where you change the heavy chain during the B cell response, is called isotype switching. And these processes are most specific for T-dependent responses, but not absolutely so. Okay? This is mainly T-dependent. And I'll come to your question in a minute. Okay, so now let's start with the response. What you should know by now is that DCs go to the T cell zone. MHC class 2 and peptide is going to activate a helper T cell. The activated helper T cell is going to expand. And those activated, expanded helper T cells are all going to induce the expression of CXCR5. And they're going to reduce the expression of CCR7. So CCR7 is the chemokine receptor that T cells use to go to the T cell zone. CXCR5 is the chemokine receptor that B cells use to go to the B cell zone. So this activated T cell, which has been activated by a DC, by peptide and MHC, has expanded, proliferated, and it's going to now have the ability, because it's changed its chemokine receptor, to migrate towards the T cell zone, because it now has a chemokine receptor that sees towards the B cell zone, towards the B cell zone, because it now has a chemokine receptor that looks like something that a B cell should have. Okay? CXCR5. At the same time, this B cell in the B cell zone, we just had this little discussion about can the B cell be activated by a monovalent antigen. So the monovalent antigen, maybe a protein antigen, comes and binds to this B cell receptor. Okay? Let's assume this is a protein, not a microbe. This step of trying to activate the B cell is not sufficient to cause this B cell to proliferate. It's not multivalent. It's not going to proliferate. It's not going to expand. But it's going to get a little bit of a kick. Maybe because of a complement-bound antigen, maybe just because of the antigen itself. And that little kick is enough for it to do a couple of things. One is it's going to induce CCR7 on the surface of this B cell. The B cell has not proliferated, but it has now suddenly become a T cell wannabe. It's got CCR7 on the surface, and this B cell starts to migrate now towards the T cell zone. So the T cell activated T cell goes towards the B cell zone. The slightly activated B cell starts to migrate towards the T cell zone. And somewhere in between, they're going to meet. Yeah? Yes? Yes. They haven't proliferated. So just for simplicity, I've shown only one T cell here. But let's say there's a huge number of T cells here. They're all moving now towards the B cell zone. Okay? The DCs, there are plenty of DCs. They can find that rare T cell and expand it. Okay? That rare T cell has been expanded, and now all of these guys are migrating towards the B cell zone. The rare B cell that saw its antigen was triggered, and it's now moving towards the T cell zone, and somewhere in between, a specific B cell will meet one of those T cells. Right? Yes?
Go back to the dendritic cell, which starts the process. Where is it exposed to the antigen?
Okay, so the dendritic cells. So if you have a cut in your skin, you have a little inflammation, you activate your mast cells and your macrophages first and so on, but you also activate DCs. The DCs capture the antigen from the microbe and go to the nearest draining lymph node, which would be the axilla. And DCs have CCR7, and so they always go to the T cell zone. So the DCs have gone to the T cell zone, and they've activated T cells, and you get T cell proliferation. That's how it all starts. Okay? Yes?
Same antigen?
It's the same antigen. We'll talk about the epitopes. It's the same antigen. Question? Say that again? Okay, so let me clarify this. I'm not sure that you guys have got what you should have got yesterday, but let me just clarify this, okay? So the antigen that is picked up by the DC, let's say it's a protein antigen, is going to be chewed up into bits. Peptides are going to be generated. They're linear peptides. They're going to bind to MHC class II molecules, and they're going to go to the surface of the DC, right? And so what the T cell sees is always a linear peptide on an MHC. The B cell could have been activated by a conformational determinant on a protein, could have been activated by a linear determinant on a protein, could be activated by anything. B cells are not discriminatory. They see everything. Okay? That B cell that's activated, if it's a protein antigen, can also internalize that protein antigen like a DC, chew it into bits, and make peptides. I'm going to come to that. Okay? We're going to come to that, but I hope you get this. Yes?
So is it the same process for the autoimmunity? Or for any autoimmune process with the cell?
In an autoimmune process, if it's a protein antigen, it's the same thing. If it's a protein antigen, it's a protein antigen, it's a protein antigen. Okay? So, yes? This priming, no. All P and T cell activation occurs in lymph nodes. The design of the system is that antigen goes to nodes. And the naive cells don't go to the tissue to find antigen. Naive cells only go to nodes. Through the lymphatics. DCs or lymphatic directly. Right? Yes, there's one more.
So what the B cell presents and what the T cell presents?
Same thing.
The linear fragment.
Identical linear fragment on the same MHC molecule.
So it will be same amino acids.
Exactly the same. It's the same MHC molecule. The D cell, the B cell, and the dendritic cell have the same MHC molecules. And they process the protein the same way. It's the same peptide. We'll come to that. I hope we get to the bottom of this as we go through. Any other questions at the back? No. Okay, so this is just the beginning, right? So, yes. I think you want to understand this really well, because if you don't get it, you missed most of immunology. Yeah. So the B cell can present, yeah, so the question is, the B cell can take different antigens up. In the case of this B cell, the one B cell you're thinking of, it's going to be specific for only one antigen. It has a receptor for only one bump on a protein. And that bump, let's say we call it a yellow triangle on a protein. This B cell is only for yellow triangles. So if the protein doesn't have a yellow triangle on its surface, the B cell is not going to take anything in. So only one B cell will process that. There may be another B cell that sees another bump on that protein, another B cell, and it will process the same peptides. Yeah, but no, even though a B cell can make several peptides from one protein, only one or two of those peptides probably bind to MHC class II. So from a given protein, you may have only one or two determinants that can bind that individual's MHC class II. For that specific peptide MHC complex, yeah, probably only one T cell. Okay? I hope I'll bring this all together in the next few slides, but interrupt me if you don't get it. Okay, so this is again going back to the process. The protein antigen can be taken up by the B cell. So there was some bump on this protein antigen was seen by the B cell receptor. It was internalized. It was chopped into peptides. The brown thing over here is the peptide. The peptide can then bind to MHC class II and be taken to the class surface. And this class II bearing peptide is now seen by the activated CD4 cell. Okay? I'll go through a few more slides to explain this. So how did the process start? You first had T helper cells being activated by DCs and expanding. These expanded T helper cells, called effectors, are going to migrate towards the B cell zone, okay, because they induced CXCR5. A specific B cell shown over here got tickled by antigen, didn't expand, and moved towards the T cell zone. This is the first T-B interaction. The first T-B interaction allows the activated CD4 T cell to activate this B cell. We'll discuss how in a minute. But this B cell is presenting the same peptide MHC complex that was used by the DC to activate the naive helper T cell. Okay? It's got the same MHC molecules. is the same protein antigen. The protein antigen that the DC used to activate these helper T cells is now being presented by this B cell to these T cells. T cells activate the B cells. We get something called an extrafollicular focus, where you get a little bit of B cell expansion, a little bit of short-lived plasma cell formation, and some activated B cells. Activated B cells will then activate some of these T cells to return the favor. You started with T cells activating a B cell, then B cells will activate a T cell to convert some of those T cells into a specialized subset called T follicular helper cells. The T follicular helper cells will go into the follicle and induce the formation of a germinal center, allowing one of these B cells to come in and become a germinal center, and I'm going to describe that in a minute. Okay? So the stepwise process is T cells activate B cells activate T cells, activated T cells go into the follicle, form a germinal center, and in the germinal center, selection is also going to depend on activated T cells. We'll come back to that. So just to walk you through this again, protein antigen captured by the B cell receptor by endocytosis is processed into peptides which bind MHC class II. MHC class II is expressed on the surface, recognized on the activated T cell by the T cell receptor, CD4 T cell. The activated T cell happens to have a molecule on its surface called the CD40 ligand. Only on activated T cells. You heard about it yesterday. CD40 ligand whacks the B cell through CD40. And the proliferation of the B cell that follows and the differentiation that follows is because of this stimulation. This triggering was enough for endocytosis and to induce some CCR7 to allow the B cell to go towards the T cell zone. This stimulation from the T cell to the B cell is the one that drives this to differentiate, proliferate, and so on. Okay? So again, just walking you through this again in a few steps here. B cell receptor recognize the protein antigen. Shown over here is the T cell epitope. This bump over here is the B cell epitope. This linear peptide over here is the T cell epitope. They are linked to each other physically. The B cell internalized the protein antigen, chopped it into bits in the lysosome, and yielded this protein epitope, this peptide epitope. The peptide epitope bound to class II, went to the cell surface, was recognized by the T cell receptor on the activated T cell. CD40 ligand on the activated T cell whacks CD40. Cytokines made by the activated T cell also activate receptors in the B cell. And this process causes B cell expansion and proliferation and differentiation. Okay? I'm reiterating this a few times because I just want to make sure you all get this. This is the hub of immunology, in a sense. Okay, so once again, T cells and B cells interact to give you two types of outcomes. They can early give you an extrafollicular B cell activation phenotype, where you have some expansion of B cells, some short-lived plasma cells, little bit of isotype switching, little bit of somatic mutation. Later on, and I'll go back to this, you can get a germinal center formation. And in the germinal center, you get isotype switching very efficiently, affinity maturation, somatic mutation and selection of cells. You get long-lived plasma cells, and you get memory cells. Long-lived plasma cells are the effector cells of the B lineage. Memory cells are the typical central memory cells of the B lineage. Central memory cells here go back to lymph nodes. Long-lived plasma cells go to the bone marrow and keep secreting humongous amounts of antibody all your life. Okay? Long-lived plasma cells. So the outcome in the germinal center response is to give you long-lived plasma cells, which you can consider to be the effector memory cells of the B lineage. They live forever, they make antibodies, they have effector function. You also get memory cells, which we can think of as central memory cells. They go back to lymph nodes, and they sit around waiting for antigen to come by, so you get a stronger response the next time. The long-lived plasma cells go to the bone marrow. They sit in the bone marrow, and they're kept alive there, and they keep secreting antibodies. Okay? I'm actually going to come back to all of these points again. Okay. All right, so now I'm telling you, I said that, when you have these activated B cells, they can in turn activate T cells. So now we have an activated B cell using something called ICOS ligand to stimulate a previously activated helper T cell. So DCs live for 2 or 3 days, and then they die. Afterwards, antigen presentation is dependent on the B cells over here. So the B cells now present antigen to the helper T cells, and drive their activation again. And this activation allows, through ICOS ligand and other things, allows the induction of a transcription factor called BCL6. So this helper T cell is now transformed into a lineage like Th1, Th2, and Th17 into a lineage of T cells called T follicular helper cells. And these T follicular helper cells probably have subsets of Th1, Th2, and Th17. I mean, this is the emerging view that, you know, when you want to get a Th2-like response, the T follicular helper cell that helps the B cell makes Th2 cytokines. If you want to get a Th17-like response, it's going to make Th17 cytokines, and so on. The T follicular helper cell, which secretes a lot of IL-21, is important to form the germinal center, and it helps to transform activated B cells into germinal center B cells. And to show you what that is, so I'm going to walk you through again. First, DC's activated helper T cells, activated helper T cells, then activated the first B cell. So T-B interaction. The second interaction is a B-T interaction, where the activated B cell induces the helper T cell to become a T follicular helper. The T follicular helper forms the germinal center, induces germinal center B cell formation in the germinal center. And I'll describe the germinal center in a bit. The outcome of the germinal center is long-lived plasma cells and memory B cells. Okay? Germinal center looks something like this. A dark zone and a light zone. This is the mantle zone. The dark zone is where the cells proliferate like crazy, the B cells, and they divide every 4 to 6 hours. The most rapidly dividing cells in the body, and they mutate the antibody genes as they divide. Okay, so they have this B cell receptor, and they go and deliberately screw it up in the hope that some B cells will now get a high affinity receptor. In the light zone is where selection events are going to occur. Okay? So you look at this in the schematic view. In the dark zone, the B cells proliferate, and then they come out into the light zone as centrocytes, and then they are tested. Cells called follicular dendritic cells, these are not DCs. Follicular dendritic cells hold immune complexes with the antigen on the surface, and if the B cell that comes out of the dark zone has a high affinity for the antigen, it can capture the antigen, it can be activated, it can get help from a T follicular helper cell. So this is the third interaction between T cells and B cells, where the T follicular helper cell helps the B cells which are highly mutated, which have the highest affinity receptor, and in a Darwinian process only allows the high affinity cells to survive, and all the other sister cells, the cells which mutated the receptors but weren't so high affinity, they die. So the T follicular helper cell mediates this third BT event. It's a T cell helping a B cell and selecting the B cell which has the highest affinity for antigen. Okay? It's competition. It's pure Darwinian. You have genetic diversification, the natural selection, survival of the fittest. Okay? Yes. The follicular dendritic cells have Fc receptors and complement receptors. So an immune complex of antigen and antibody would be held on the surface. And they're just holding it up and seeing which B cell really has a high affinity receptor, to be picked out. Yes? I mean, there is a threshold, but how you measure it is not clear. Clearly, the highest affinity guys live and the low affinity guys die. And you can study them by sequence and find all the mutated guys who are screwed up and the mutated guys who did the right thing and became high affinity. And those are the guys that live. Yes?
If you compare your B cell receptor to your follicular, how does the T follicular helper know? Is there
The T follicular helper was activated originally by that DC and then by that B cell, so it has a receptor for an MHC and peptide. The B cell with the highest affinity B cell receptor will capture the most protein antigen to have that peptide presented.
so the B cell is talking to both the FDC and TFH?
It's capturing antigen from the FDC and then presenting peptide from that antigen to the T follicular helper. If it has a lousy receptor, it presents very little peptide, it doesn't get help. If it has a really high affinity receptor, it presents peptide, it gets help. Yes? Beg your pardon? Short-lived plasma cells, which are generated in a T-dependent response are usually in the extrafollicular focus. They live for a few days and then they die. They don't go to the germinal center. The only cells that go to so I should explain this. Once you make the T follicular helper cell, one particular B cell goes into the follicle, is converted into an activated B cell and starts a germinal center. So each germinal center may come from 1 or 2 B cells only, which are then allowed to expand, proliferate, and then you select the highest affinity guy. And they give you, when they are done and they're selected, the highest affinity guys give you long-lived plasma cells and memory B cells. Yes?
What is the estimation to the degree of expansion of the reservoir of the antibody affinities by these processes?
So if you didn't have high affinity antibodies, you would get no protection against viruses. Any vaccine that works, as I told you, makes high affinity antibodies. All autoimmune diseases where the autoantibodies cause problem, somatically mutated antibodies.
So one can explain very much that potentially patients that lack a or the ability to fight in an efficient way infections might potentially be hampered by if the process is somewhat hampered in their case.
Hyper-IgM syndrome patients, X-linked has both T cell and B cell problems, but other ones, like the young mutation and so on, they have a defect in affinity. Yes. Question? Yes. So in receptor editing, the most efficient way to edit is to make a new light chain. Okay, but there, all you're making a new light chain, you're not mutating anything, you're just making a rearrangement. In somatic mutation, you mutate both heavy chain and light chains. And then you select for the highest affinity B cell. Question? Yeah, that's a good question. So the T follicular helper cell actually sets a tone. See, the IL-21 that's secreted makes all the B cells very prone to apoptosis. They induce high levels of FAS, they induce high levels of BIM, and they're all about to die, except for the ones that are saved by high affinity interactions. Questions? So T follicular helper cells basically are providing B cell help for class switching. And so if the T follicular helper cell makes slightly different cytokines, it will then allow the B cell to switch to a particular isotype. Th2 cytokines will give, IL-4 will give you IgE and IgG4. IL-17s give you IgGs mainly. Okay, so there's some differences. Yes? It holds the antigen through immune complexes. So it has antigen-antibody complexes are held by Fc receptors or by complement receptors on the surface. So clearly the different bump on the protein bound to a different antibody would be held up by the Fc receptor, and that would expose the antigen, let's say the yellow bump that you talked about, to the B cell that comes by. It's an immune complex being held up. Yes? How were all these discoveries made over the years? Not only by mating experiments, I guess the visual discoveries are very recent, where people have seen this by 2-photon imaging. But a lot of the discoveries have been made over the years by looking at the cells in the germinal center. The T cells that you pulled out had this phenotype. So you call them T follicular helper cells, and you characterize them, and then you made knockout mice and so on. Yeah? Okay. All right. So this is just an overview of what happens in germinal centers. Naive B cells take up the antigen, get T cell help. They induce something called AID, which I'll talk about very briefly. You get B cells that are somatically mutated. Somatically mutated B cells then have antigen presented by FDCs. Yeah, in 10 minutes, definitely. FDCs, and then T follicular helper cells provide the help to the highest affinity guys. The highest affinity B cells live. The low affinity B cells die. The high affinity B cells then give you long-lived plasma cells and memory cells. Is this clear? Yes.
How many plasma cells, I mean, the mass of the finally selected high affinity B cells does one B cell give rise to one plasma cell?
No, so the underlying principle is this, right? During an immune response, you want to make a lot of effectors, then kill most of those effectors, or make them commit suicide, and have a few cells left behind, a few more, maybe tenfold more than before, so that you have memory. So yeah, there's an increase because you have memory, but most of your cells die. That's called contraction of the immune response. Okay, so just to give you the big picture view, Abul, I'm stopping in 10 minutes according to Mark, so you can sit down, relax. I'll do a little rap before you come on. Okay? So this is just an overview. You get T cell help. The T cell makes cytokines. Depending on the cytokines made by the T helper cell, the T follicular helper cell, the B cell will switch to different isotypes. If it's IL-4 that's being made here by the helper T cell, if it has a Th2 phenotype, you're going to make the IL-4 is going to induce the opening of IgE and IgG4 loci. So the class switching, which I'll describe in a minute, is going to go from IgM to IgE or IgG4. So depending on the cytokine, you're going to switch to a different isotype so that you're tailoring the T cell help makes the B cell produce the kind of antibodies that are going to be most helpful in the context of the antigen that started it all. So the antigen, the type of pathogen induces the type of helper T cell. The type of helper T cell makes certain cytokines. Those certain cytokines induce a type of class switch which is appropriate for that pathogen. So if this was a worm, you would make IgE. Okay? So class switching very quickly. So you have the VDJ exon over here in the heavy chain, and then you have the constant region of mu shown in this green bar. Way downstream is, let's say, the epsilon locus for IgE, where you have the constant region of epsilon over here. Now, upstream of this constant region, there's a switch region, this circle over here. And upstream of that is a promoter called the I epsilon promoter. So the goal would be that if this B cell got help from cytokines like IL-4, that IL-4 will induce nuclear proteins which will bind to this promoter over here and induce transcription of the epsilon locus. So you're going to open up the epsilon locus transcriptionally in order for the cell to decide to switch from IgM, so this was mu, from IgM to IgE. Now, if you looked at the locus, this is your locus. Mu is miles upstream of epsilon. But what you want to do at the end of the day is to cause a break in the switch mu region, to cause a break in the switch epsilon region. So you bring them close together, you cause breaks, and then you're going to join the switch mu region to the switch epsilon region. And so that VDJ exon, which was near mu, is now being placed near epsilon. So the cell that made IgM will now be a cell that makes IgE. So basically, it's a DNA recombination event, different from VDJ recombination, but it involves double-strand breaks here, double-strand break there, cytokine-induced breaks. The cytokine is choosing the locus. It says, go to IgE, please. And so this cell is going to switch now from mu to epsilon, and it's going to make the DNA, make the RNA, and it's going to make the protein. Okay? Somatic mutation just means that after you activate the cell, in the CDR regions, as you see as days of activation go by, you get more and more mutations in the heavy and light chains. And then you get an antibody of higher and higher affinity. Okay? So you started with a low affinity antibody, where there's a round something in a square peg or whatever, the other way around. Okay? And then you mutate the antibody through somatic mutation and selection. Now you have an antibody that really tightly fits the shape of the antigen because it acquired mutations in the V regions. So that's affinity maturation. Somatic mutation, selection to give you high affinity antibody. Driving this process is an enzyme. I'm going to skip these slides in the interest of time. This activation-induced cytidine deaminase. If you have questions, I'll answer them later. Okay? But basically, this enzyme converts Cs to Us in DNA and mediates both class switching and somatic mutation, and is somewhat understood. Okay? So I'm going to skip these and tell you that in in isotype switching, it's driven by AID, and it's transcription dependent. But I haven't gone through the details, so unless you have questions, I'll skip this. So just to tell you that, yes, even T-independent switching occurs. It's not only T-dependent. Some T-independent switching does occur. CD40 drives AID induction, but there are other ways in a T-independent manner through a receptor called TACI that you can also induce AID. Okay? And finally, you have terminal differentiation to plasma cells. There are cytokines involved in this process. There's a growth factor receptor on plasma cells that keeps them alive. It's called BCMA. So there are different transcription factors in B cells, germinal center cells, plasma cells. I won't belabor the point. Just to remind you again that BCMA is that receptor on plasma cells that uses BAF and APRIL in the bone marrow to keep these cells alive. Okay? There are a lot of changing notions. I'm going to skip this tolerance part and go right to the end. Okay? So I mentioned this before. There's receptor editing and so on. There are different forms of B cell tolerance. In the periphery, you can have anergy. You can have something called clonal ignorance that I'll mention briefly. You can still have deletion. And you can have Tregs causing B cells to become tolerant. Okay, so there's different ways in which tolerance can be made in the periphery. Centrally, I said mainly receptor editing. And clonal ignorance just means that if I have a B cell that is weakly self-reactive, I want to keep it in the follicle and not let it go to the T cell zone. Because if it went to the T cell zone, it will get T cell help, and a weakly self-reactive cell may become strongly self-reactive. So there needs to be a squelching process to keep weakly self-reactive B cells in the follicle and prevent them from inducing CCR7 and going here. So there's a threshold being set. Only if you cross a threshold are you allowed to go to the T cell zone. And that process by which there's a mechanism by which this is mediated, which I'm skipping over, which basically keeps B cells tolerant. It keeps them in the B cell zone, preventing them from going towards the T cell zone, because if they went there, they would get T cell help and become somatically mutated. That would be a problem. Okay, the inhibitory receptors on B cells. So one example just glossed over was CD22. There's another receptor called Fc gamma R2b, which we mentioned before, which is also an inhibitory receptor in B cells. And so if you think finally in the last two slides, what happens in terms of B cells and autoimmunity. So clearly it seems to be that some rare genetic variants change the function of key immune proteins in B cells, T cells, innate immune cells, and so on. Common genetic variants, more than changing functions of proteins, they do that sometimes, mainly seem to change the expression patterns of certain genes. So common variants are often in enhancers of genes. So these also contribute to, altered gene expression from one individual to the other. And then some microbes play a role in the process. Commensals, pathogens can contribute from the environmental point of view to inducing autoimmunity. And next-gen sequencing is revealing a lot of this information. Maybe this will be covered tomorrow. Okay, I'm skipping this. Okay, so finally, therapeutic strategies. Rituxan, very helpful. You deplete B cells. It's not just because you get rid of antibodies, because you also get rid of CD4 memory cells. If you deplete B cells, CD4 memory cells, which might be causing your multiple sclerosis or causing your type 1 diabetes, they're also not going to be able to survive. And that's probably why Rituxan works. IVIG induces Fc gamma R2b, the inhibitory Fc receptor. BAF antagonists like Benlysta have not been that good therapeutically. They don't do as much as Rituxan. So they do block some B cell function and B cell development, but they don't get rid of memory cells. So you have a lot of memory left behind. So clinical trials with Benlysta have not been so effective so far. Okay? Very excitingly, signaling inhibitors, BTK inhibitors. BTK is that kinase downstream of the B cell receptor, which is mutated in X-linked gammaglobulinemia. SYK inhibitors. SYK is that kinase, again, downstream of the B cell receptor. These are now in clinical trials with fantastic results in autoimmune patients. So blocking B cell function by using small molecules might one day replace using biologics, like removing B cells altogether. It's possible. Okay? But these are very promising right now. And you've probably heard of JAK3 inhibitors and other small molecules. Okay, this is the point I wanted to make, that B cells, activated B cells, are required for CD4 memory. That's why maybe Rituxan really works. And so this is my last slide before I do a little rhyme that is going to get Abul happy and shaking his hips. Okay? You start with resting B cells. When you have T cell help, let's go back. When you have T cell help, protein antigens are involved in calling in T cell help. You get helper T cells come in, provide stimuli, you get clonal expansion, you get switching, you get somatic mutation, and affinity maturation selects the high affinity cells, you get memory cells, and this is the hallmark of a full-blown immune response driven by T cell help. Okay? Yes? We don't know that. How is the decision made to make a B cell into a plasma cell or a memory cell? That's not very clear. We know what transcription factors are turned on, but what signal makes it go this way or that, it's not very clear. Yes, so a B cell always starts with IgM and IgG. It could go to an IgG isotype, which is upstream of IgE. It could then be induced to switch to IgE, given the right cytokines, by making one more rearrangement. So a memory cell, which is IgG, in the lymph node, next time antigen comes by, if the cytokines are different, and it's a worm or whatever, it can drive it maybe to switch to IgE. Everything occurs in lymph nodes or in secondary lymphoid organs, Peyer's patches, lymph nodes, spleen. Yes? Question? Yes? So the question is, do antigen-activated B cells activate naive T cells? So the T cells, the B cells activate naive B cells, right? The B cells I'm showing you are virtually naive, right? The naive B cell sees antigen, moves to the T cell zone, gets help from an activated T cell. So that is a naive B cell. A naive B cell does not activate a T cell. So typically a naive B cell, you would think, would not be able to activate a T cell because of multiple things. Totally naive B cell has to come to the T cell zone. Something has to bring it there, right? To activate a T cell, the naive B cell has to be expanded and activated through T cell help, and that is going to require something else. So a naive T cell will be activated by a DC. A naive B cell will be activated by a T cell. I think that would be, if I had to make a rule. No, a naive T cell is sitting in the T cell zone. It is typically activated by a DC. Because the chances are that the antigen will be taken up by DC before it goes to a specific B cell. Right? So I would always think of naive T cell is activated by DCs. Naive B cell is activated by activated T cells. Is that clear? Yes. So if I have a weakly self-reactive cell, right? So then it didn't get a strong stimulus in the bone marrow. It didn't have a high affinity receptor. So that was the kind of cell that I allowed to differentiate. Everything is slightly self-reactive. It differentiates. So you want that B cell to be activated by a foreign antigen, which has a high affinity for it, not by the self-antigen that can just tickle it. So you want to keep it quiescent and not come out of the B cell zone, because if it did, then its only function is to respond to foreign antigens. But things go wrong sometimes, right? And its only function is to see a foreign antigen. That would be a high affinity foreign antigen. Okay, yes, sorry. Slightly, yes. Well, it's not, yeah, you could call it selection, but no expansion, right? So it's just been slightly activated by antigen and allowed to move out. So strictly speaking, just slightly activated. Yes? So, you know, many vaccines, many polysaccharides, first of all, it's not absolute, right? So some polysaccharides can activate responses that look like T-dependent responses, first of all. Secondly, you have these conjugate vaccines, where you conjugate the polysaccharide to a protein to get a really good T-dependent response, to get long-lived responses. So the best way to do it is with protein. So conjugating the polysaccharide to protein makes it better. But LPS and many TLR activating polysaccharides and so on can induce fairly good long-term memory. So it's not absolute. Yes. Sorry, I'll come to you. Fetal liver. All hematopoiesis starts in the fetal liver. When we make our lymphocytes, we start first near the aorta in the fetus, and then it goes to the fetal liver. That's where hematopoietic stem cells come from. Then you go to the bone marrow only after you're born. The regular liver doesn't have hematopoiesis. The fetal liver has hematopoiesis. Okay? Okay, I'm wrapping it up with a wrap. Okay. Okay. Okay, I'll answer all your questions in the afternoon if you have more questions, in that famous session that no one's coming to. Okay? Okay, this is especially for Abul. Abul, you move your hips over there, okay? Okay. Pathogens lurking everywhere. Go get them first. They never fight fair. Give me a D, give me a J, put a V in frame, it'll be A-OK. Surrogate light chains, pre-TCR. Kappa or alpha can't be too far. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Naive cells dance from hall to hall, pining and hoping to get that call. Looking for antigen below and above. Many will die of unrequited love. Thread that peptide into TAP. Everybody do the lymphocyte wrap. DCs lurking everywhere. They'll get your antigen, don't despair. CCL21 to CCR7. Come in, DC baby, make this lymphocyte heaven. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Wake up, T cell, activate ZAP. T cell says takes 2 hands to clap. Give me signal 1, give me signal 2, and I'll make you a cytokine brew. Thread that peptide into TAP. Everybody do the lymphocyte wrap. Collectins, complement, macrophages. That's what you get in the early stages. Then antibodies, cytokines, CTLs too. Hey, pathogen, that's the end of you. Thread that peptide into TAP. Everybody do the lymphocyte wrap. I'm now going to introduce Abul Abbas, who will move his hips for you.